Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about vaccinations and vaccines. Two artificial methods exist to make an individual immune to a disease. The first one we will talk about is the active immunization. Active immunization is administered when a vaccine is introduced into a patient and the patient actively mounts a protective immune response. So in this picture, we see a president getting a active immunization. He's getting vaccinated. That vaccine will then go into his bloodstream where an antigen presenting cell will consume the vaccine. It will then process the specific subunit of protein, most likely. It will present that protein on the surface of the cell, the antigen presenting cell. Then a T cell, a naive T cell, will then bind or interact with the antigen presenting cell. They will communicate. Then the T cell is going to send a signal to B cells, and then B cells are going to produce antibodies. So this is a process that will allow the individual patient to generate antibodies through an active immunization. The second example is passive immunization. Passive immunization is when an individual acquires immunity through the transfer of the antibodies from an individual that was already immunized. The immunized individual builds the antibodies and you take the antibodies from that immune individual. That immune individual can be a horse or some type of animal, or it can be a human. So that is the difference between active immunity and passive immunity, or active immunization, where you build your own antibodies, and passive immunization, when you take someone else's antibodies. Vaccinated individuals can protect those around them from getting the infection. This is referred to as herd immunity. Not everyone in a community needs to be immune to prevent the spread. If a high enough proportion of immune people are created, the majority will protect the minority. The minority might be the few that are susceptible because of a weakened immune system. If the majority gets immune, then they will prevent the spread to those individuals that cannot get the immunity. If you look at this picture, the red square highlights one that I want to talk about. You have the red circles that are infected. The white circles are healthy individuals that were not vaccinated. And the blue circles are healthy individuals that were vaccinated. So if you look at the red one all by itself to the far right top corner, that infected individual can only spread it to one person, that healthy unvaccinated person. Right beside that healthy unvaccinated person is a vaccinated person. So the spread stops there. It can't move any further. So again, if you have the majority of the individuals vaccinated or immune, then it's hard for the virus or pathogen to spread. This is called herd immunity. We will discuss three types of vaccines. The first one we'll look at is the whole pathogen. We will spend the entire talk on that. Then on a subsequent PowerPoint, I'll discuss subunit vaccines. And then on the last PowerPoint, I will discuss nucleic acid vaccines. So the first one is whole pathogen. We're not going to cut it up. We're not going to give you parts of it. We're not going to give you a subunit or nucleic acid. We're going to give you the whole pathogen. So here's my concept map to show you where we're going. Today, we will only talk about the whole pathogen. The whole pathogen can be broken into three subgroups. The first one is a live whole vaccine. Then we'll talk about a killed vaccine. And then we'll talk about a live attenuated chimeric vaccine. The first one is living, but it has been modified. The modification reduces virulence. So when we modify it, it's called attenuated. So attenuated live vaccines emerged in the 1950s with advances in tissue culture. Attenuated or weakened pathogens are active but have reduced virulence. They have been weakened by repeatedly subculturing them. Since they are weakened, they cannot cause the disease. Viruses are often attenuated by raising them in tissue culture cells for which they are not adapted for. Therefore, the constant replication in tissue culture that it is not prepared for, the virus will lose its ability to produce the disease. We therefore weaken the virus. This can be done by growing it in an egg. If you look at a lot of the vaccines contain egg. Now you understand why. To create a less virulent form of the microbe, sometimes we have to grow it in an egg. The original 1954 measles virus was transformed to a weakened state by performing an exhausting 10-year study where they did serial passages using tissue culture media. So this virus was grown in tissue culture media that it was not able to effectively grow in. Therefore, it weakened the virus every time it replicated. And this took place for 10 years. And after 10 years of replication in this tissue culture, we ended up with a weakened strain that we could use as a vaccine. Bacteria can be made avirulent by culturing under unusual conditions or through genetic manipulation. One type of genetic manipulation is by removing a gene that causes virulence. In this picture, we are removing the gene that causes virulence, and now to the far right we have a harmless attenuated live pathogen. A relatively small dose of attenuated vaccine is needed to be administered. Once we have these few microbes that have been injected into your body, then they will start to replicate. Your immune system will attack this attenuated virus. So the attenuated virus can result in a mild infection, but not the actual disease. These mild reactions or these mild infections are called adverse reactions. Viral vaccines trigger a cell-mediated immune response, which is dominated by T helper cells and cytotoxic T cells. So by using a live attenuated virus as a vaccine, you're going to initiate a different type of immune response. It's called a cell-mediated. Some of the cells involved are the T helper and the T cytotoxic. We do have a few problems with attenuated viruses or attenuated vaccines. The first problem is attenuated microbes may retain enough virulence to cause disease, especially in immune suppressed individuals. So since their immune system is already weakened, this living virus or this living microbe might still harm them. The second problem is pregnant women should not receive live vaccines due to the risk of the modified pathogen crossing the placenta. And the third problem is modified viruses may occasionally revert back to the wild type, or they might mutate to a virulent form. Here was one of the problems seen with the oral live polio vaccine. It is known to be able to revert back to a disease-causing form. Remember, this is a live organism that once you consume or inject, it is able to replicate. If it is able to replicate, it might revert back or it might mutate to a active virulent strain. So that is one problem seen with these live attenuated vaccines. Here are some examples of live attenuated vaccines. Even though a small dose is needed with attenuated live vaccines, a small percent of recipients do not respond to the first dose of the living vaccine, and a second dose is recommended to provide high levels of immunity. This is seen in a few of those live attenuated vaccines, and one example is the MMR vaccine. So we're done with live attenuated. Now we will look at killed. The killed vaccine is going to be inactivated. Inactivated vaccines were discovered to induce immune response in the 19th century, according to Western medicine. But we now know that the Africans and the Asians were experimenting with this hundreds of years prior. Common inactivation occurs through chemicals, heat, or radiation. One example of inactivation is with the herpes A vaccine. This virus is inactivated by using formalin, and this was discovered in 1995. So this is a killed virus that was killed using formalin. Formaldehyde is commonly used to inactivate microbes by cross-linking their proteins and nucleic acids. So it modifies the virus in such a way that it reduces the virulence. During inactivation, the antigens responsible for stimulating protective immunity must not be altered. So when you are killing this virus, you want certain proteins, certain antigens to not be affected. You want those antigens to still be available so that you can appropriately build an immune response against them. So this unaltered exogenous antigen will stimulate the T helper cell that will then promote antibodies to be released from the B cells. So if you somehow alter this exogenous antigen, this protein, if you alter it, then the T helper cell will not be able to help produce the antibodies. Inactive vaccines are safer than live vaccines since they cannot replicate or mutate to a virulent form. Therefore, immune suppressed individuals can acquire this vaccine because it is dead. The entire dose of antigen, however, must be administered since the kill pathogen cannot replicate. This type requires multiple doses since they are not replicating. So with the living virus, you can give the patient a little bit, a little small injection, because that virus will replicate. But since this virus is dead, since this pathogen is dead, you have to increase the amount of antigen you are putting into the person. The response is mostly humoral. B cells are responding. Little to no cellular immunity is initiated. So you're not activating the T helper cells. And this, when we use these dead viruses or these dead vaccines, the antibody titers against inactive antigen diminishes with time. Periodic increase or booster shots will be required. So if you've ever experienced a booster shot, you most likely was given a lesser type of vaccine, a vaccine that was dead, that was not able to replicate. So therefore you had to have a larger dose of the antigen to develop this immune response. Here are some examples of inactivated vaccines. These inactivated vaccines are no longer available in the US. We do have some problems with inactivated vaccines. The whole agent vaccine may stimulate an immune response due to non-antigen portions of the microbe. We already mentioned this. Since the microbe is dead, it is going to be weaker than some of the others because it is not able to replicate. These dead vaccines must be administered in high or multiple doses, and both may produce allergic reactions. To increase efficacy, the incorporation of adjuvant can be used. And adjuvants may also stimulate local inflammation. And listed in this table, I have several adjuvants. So now we're going to look at some live attenuated chimeric vaccines. Here is the concept map. We're on the final subcategory of whole pathogen. This type of vaccine contains a mixture of different biological molecules from parent viruses. In this picture, we see an attenuated vaccine from strain SA14-142, and we have another one, strain 17D yellow fever. So chimeric is when you combine organisms. So this is a recombinant chimeric JE vaccine virus. This is a mixture of two different biological molecules from parent viruses. The National Institute of Allergic and Infectious Disease has recently merged a dengue virus backbone with a Zika virus surface protein to create a chimeric whole vaccine.